The human immune system is a highly complex network of millions of immune cells that protect the body from constant exposure to pathogens and disease. One defense mechanism that has evolved over time is the body's response to yeast. These potentially harmful organisms are initially the target of antibodies, which bind to the pathogen and attract soluble blood proteins called complement to also bind to the target. The complement protein now attracts neutrophils, the most abundant immune cell in the body. Neutrophils are innate immune cells that are part of the body's first line of defense. Special receptors on their surface bind to complement-coded targets. These receptors have a second site that detects and binds beta-glucan, a carbohydrate found in the cell walls of yeast. Both the complement and the beta-glucan binding sites must be occupied for the neutrophil to recognize the yeast cell as a non-self pathogen and kill it. Innate immune cells, like neutrophils, are the body's primary response to foreign challenges. Biothera is developing patented beta-glucan compounds that significantly enhance the immune system to fight a wide range of diseases. Biothera's pharmaceutical compounds are administered intravenously. Applications include pharmaceuticals, functional foods, and animal feeds. When taken orally, beta-glucan passes through the stomach and is the right size for uptake through the Peyer's patches, which are specialized regions in the small intestine. In the Peyer's patch, M cells pick up and transport the beta-glucan particles into the body and present them to innate immune cells called macrophages. Similarly, the injected beta-glucan is engulfed or phagocytized by macrophages in circulation. From this point on, the mechanism of action is very similar for both the oral and injectable beta-glucan. The macrophages transport the beta-glucan to immune organs throughout the body via the lymphatic system. While in the immune organs, the macrophages digest the beta-glucan into smaller fragments and slowly release them over a number of days. These fragments bind to neutrophils via the complement receptor, priming them for activity. Activated by beta-glucan, the neutrophils now more quickly navigate to the site of a foreign challenge, a process known as chemotaxis. With the beta-glucan already present, neutrophils need to simply bind to the complement on a bacteria or virus cell to activate the enhanced killing ability of these immune cells. Neutrophils, however, are normally not involved in the fight against cancer because they do not recognize this abnormal human tissue as non-self. When antibodies bind to cancer cells and attract complement, these primed neutrophils now see them as non-self and begin killing them as if they were yeast. The result is a novel therapy that significantly increases the efficacy of monoclonal antibodies. This combination therapy engages a new arm of the immune system into the battle against cancer.